Chapter 3. The mouth as the first organ of digestion. Digestion begins in the mouth because the system requires early transformation and signaling before material reaches the stomach. The mouth is not a passive entry point. It is an active interface where mechanical reduction, fluid mixing, and neural activation converge to prepare the entire digestive tract for incoming food. What happens here determines how effectively the system can initiate the sequence that follows. When this stage is performed with sufficient engagement, downstream processes receive material that is already partially processed and a signal that digestion should proceed at full capacity. When it is bypassed or compressed, both the material and the signaling are degraded from a start. Mechanical breakdown is the first transformation. Chewing reduces particle size and increases surface area, allowing digestive fluids to interact with food more effectively. Larger particles require more intensive chemical processing later, placing a greater demand on gastric acid and enzymatic activity. When chewing is thorough, the stomach receives material that is easier to penetrate and alter. This reduces the workload required to unfold protein structure and allows enzymatic processes to proceed more efficiently. Mechanical reduction is not separate from chemical digestion. It sets the conditions under which chemical digestion can occur. Saliva introduces the first layer of chemical processing while also altering the physical properties of the food. It contains enzymes that begin the breakdown of certain macromolecules and fluids that hydrate and disperse particles into a cohesive mass. This hydration improves the ability of the stomach to mix and expose material to acid. Without sufficient saliva, food enters the stomach in a drier, less integrated form that resists uniform exposure to digestive fluids. The result is uneven processing, where some portions are altered effectively and others remain structurally intact. The mouth also serves as a signaling center that activates the digestive system before swallowing occurs. Sensory input from taste, smell, and texture triggers neural pathways that increase salivary secretion, stimulate gastric acid production, and prepare the pancreas and liver for incoming material. This anticipatory phase allows the system to align output with input. When food reaches the stomach under these conditions, acid production is already increasing, and the system is positioned to begin chemical transformation immediately. When this signaling is weak or absent, the stomach must react after food has arrived, creating a delay that reduces the effectiveness of early processing. The quality of this signaling depends on engagement within the mouth. Rapid eating, minimal chewing, and limited sensory exposure reduce the activation of these pathways. The system receives less information about the incoming material and generates a weaker preparatory response. This leads to lower acid output, less coordinated enzyme release, and reduced bile signaling. The sequence begins with a deficit that carries forward into every subsequent stage. The stomach becomes a reactive environment rather than a prepared one, and downstream processes are forced to compensate. Swallowing marks the transition from preparation to processing, but the quality of that transition is determined by what occurred in the mouth. A well-formed bolus, adequately hydrated and mechanically reduced, moves into the stomach as a substrate that can be efficiently transformed. A poorly prepared bolus arrives as a collection of larger, less integrated particles that require additional processing under conditions that may already be constrained. The difference at this stage influences the entire sequence, as each downstream step inherits the properties of the material it receives. Treating the mouth as the first organ of digestion reframes the starting point of the system. It establishes that digestion does not begin in the stomach, and that early-stage preparation is not optional. Mechanical breakdown, salivary processing, and neural activation form the initial conditions that determine how effectively the system can operate. When this stage is restored, the sequence begins with alignment between input and capacity, and the burden placed on downstream processes is reduced. Cephalic phase activation. The cephalic phase is the initial activation of digestion driven by sensory input before food reaches the stomach. Sight, smell, taste, and even the expectation of eating trigger neural pathways that increase secretory output across the digestive system. This phase prepares the stomach, pancreas, and liver to receive incoming material by elevating acid production, priming enzyme release, and initiating signals that will coordinate bile flow. The system does not wait for food to arrive and then react. It anticipates and adjusts its output in advance, aligning capacity with the expected load. This activation is mediated primarily through parasympathetic signaling. Sensory input is processed centrally and transmitted through pathways such as the vagus nerve to digestive organs, where it directly influences glandular activity. Gastric parietal cells begin increasing acid secretion. Chief cells prepare to release enzymes, and the pancreas and gallbladder are primed for coordinated output. The result is a system that is already moving toward full operational capacity by the time swallowing occurs. The first material that enters the stomach encounters an environment that is prepared to alter its structure immediately. The strength of this phase depends on the quality and duration of sensory exposure. Extended interaction with food in the mouth increases the intensity of neural signaling. Taste receptors are stimulated more fully, olfactory input remains active, and the mechanical act of chewing reinforces the signal that digestion is about to proceed. When this exposure is sufficient, acid production rises in parallel with intake, reducing the lag between arrival and processing. When exposure is brief, as in rapid eating, the signal is weaker and shorter. The stomach receives food before acid output has reached effective levels, and the sequence begins with reduced alignment between input and capacity. The cephalic phase also influences downstream coordination. Signals generated at this stage contribute to the timing of pancreatic enzyme release and bile delivery. These processes are not isolated reactions, but part of a coordinated sequence that begins with early activation. When the cephalic phase is strong, enzyme secretion and bile flow are more likely to align with the arrival of partially processed material from the stomach. When it is weak, these outputs become less synchronized and the system loses precision. The small intestine receives material that has not been fully prepared and must operate under conditions that favor compensation rather than efficient processing. Disruption of this phase does not eliminate digestion, but it lowers the starting point of the entire system. Each subsequent stage begins from a position of reduced output, and the effects accumulate. Proteins are not fully unfolded, enzymes encounter resistant substrates, fats are not consistently emulsified, and absorption becomes less efficient. These changes are often attributed to later stages, but they originate at the level of early activation. The sequence is misaligned from the beginning. Restoring cephalic phase activation increases capacity before the first transformation occurs. Slower eating, thorough chewing, and sustained sensory engagement extend the duration and intensity of neural signaling, allowing secretory output to rise in advance. The stomach receives material under conditions that support immediate processing, and downstream systems operate on substrates that have been properly prepared. The sequence regains alignment because the system has been activated before it is asked to perform. Sensory signaling and digestive preparation. Sensory input sets the initial conditions for digestion by informing the system that food is about to enter and by specifying the type of material that will need to be processed. Taste, smell, texture, and visual cues activate neural pathways that increase secretory output and coordinate timing across digestive organs. This signaling occurs before swallowing and establishes the level of readiness within the system. When it is strong, the stomach begins increasing acid production, the pancreas prepares for enzyme release, and bile flow is primed to align with incoming fat. The system does not wait for physical contact with food in the stomach to begin these processes. It prepares in advance so that the first stage of chemical transformation can occur immediately. The intensity and duration of this signaling depend on engagement within the mouth. Prolonged exposure to taste and texture provides continuous input that reinforces neural activation. Chewing extends this exposure, allowing signals to build rather than peak briefly and decline. As this occurs, salivary secretion increases, and early enzymatic activity begins to alter certain components of the food. More importantly, the central control of digestion adjusts its output upward, ensuring that gastric and pancreatic responses are scaled to match the anticipated load. When sensory engagement is limited, these signals are reduced. The system receives less information and generates a weaker preparatory response, leading to lower output at the start of digestion. This preparation directly affects the efficiency of gastric processing. When sensory signaling is sufficient, the stomach receives food under conditions where acid production is already rising. Proteins encounter an environment capable of altering their structure early, which facilitates subsequent enzymatic breakdown. When signaling is weak, acid output lags behind the arrival of food. The stomach must increase secretion after material has already entered, creating a delay that reduces the effectiveness of the initial transformation. The material that leaves the stomach reflects this delay, carrying more structural resistance into the small intestine. Pancreatic and biliary coordination also depend on early signaling. The release of enzymes and bile is timed based on input that begins in the cephalic phase. When this input is strong, enzyme secretion aligns with the arrival of partially processed material, and bile is delivered in a pattern that supports fat emulsification. When input is weak, these processes lose synchronization. Enzymes may be released in insufficient amounts or at the wrong time, and bile may not be available when fats require organization into absorbable structures. The system continues to function, but it does so with reduced precision and increased variability. Sensory signaling therefore acts as a control input that determines how the sequence of digestion is initiated. It does not change the composition of the food, but it changes the system's ability to process that composition. Strong signaling creates alignment between input and capacity, allowing each stage to operate with sufficient output and timing. Weak signaling lowers the starting point of the sequence, forcing downstream systems to compensate for incomplete preparation. The difference in outcome is established before the stomach begins its work, based on how effectively the system was prepared to receive what is about to arrive. Mechanical breakdown and signaling integration. Mechanical breakdown in the mouth is not a separate step from chemical digestion. It is the first transformation that determines how effectively all subsequent transformations can occur, and it operates in parallel with neural signaling that prepares the rest of the system. Chewing reduces particle size, increases surface area, and distributes saliva throughout the food matrix, but at the same time it amplifies sensory input that drives parasympathetic activation. The physical act of breaking down food and the neural signaling that coordinates digestion are integrated processes. When one is reduced, the other is weakened, and the sequence begins with both structural and signaling deficits. Particle size directly affects how digestive fluids interact with food. Smaller, well-chewed particles expose more surface area to gastric acid, allowing proteins to be unfolded more uniformly and efficiently. Larger particles resist penetration, leaving internal portions structurally intact even as outer layers are partially processed. This creates uneven substrates that move through the system with variable levels of preparation. Enzymes encounter these substrates with reduced efficiency because access to cleavage sites is inconsistent. Mechanical reduction in the mouth determines whether the stomach is working on a uniform substrate or a fragmented one with areas that remain resistant to change. Saliva integrates into this process by hydrating and dispersing particles into a cohesive bolus. This hydration allows gastric mixing to occur more effectively, ensuring that acid can contact the entire mass rather than isolated regions. Without sufficient saliva, food enters the stomach in a drier, less integrated form that limits uniform exposure to digestive fluids. Some portions may be adequately processed while others remain largely unchanged, increasing the variability of material that moves forward. The stomach must then compensate for this inconsistency, often by prolonging retention or passing partially processed material into the intestine. At the same time, chewing reinforces sensory signaling that drives digestive preparation. Each cycle of mastication stimulates taste receptors, maintains olfactory input, and sends continuous feedback to the central nervous system. This sustained input increases parasympathetic signaling, which in turn elevates gastric acid production and primes pancreatic and biliary output. Mechanical breakdown therefore functions as a driver of signaling intensity. The longer and more thoroughly food is processed in the mouth, the stronger the preparatory response becomes. When chewing is minimal, both the physical reduction of food and the signaling required to prepare the system are diminished simultaneously. This integration affects the timing of the entire sequence. When mechanical breakdown and signaling are aligned, the stomach receives a well-prepared bolus under conditions where acid production is already increasing. The initial transformation occurs without delay, and downstream processes receive substrates that have been uniformly altered. When this alignment is absent, the stomach must process larger, less hydrated particles while also increasing acid output after the fact. The sequence begins out of sync, and that misalignment propagates through enzyme release, bile flow, and intestinal absorption. Mechanical breakdown and signaling integration establish the first level of coordination in digestion. They determine both the structural state of the incoming material and the readiness of the system that will process it. When this stage is executed with sufficient engagement, the system begins with alignment between input and capacity. When it is reduced, the system starts with uneven substrates and weakened signaling, forcing every downstream step to compensate for conditions that should have been resolved at the beginning.